So, hmm, as you all know, intermediate exams are about to start. So, let us study questions from second year. Okay, second year botany part. Okay, so let us start. Hmm, Just give me a moment, please. Okay, so today we will be learning second year concepts. So, in second year, we will start with principle of inheritance and variation part. Okay, because plant growth and development we are done, bacteria questions we are done. Okay, so let us start with principles of inheritance and variation. Okay, now let us start. Okay, so our first question is, what is the cross between F1 progeny and the homozygous recessive parent is called? How is it useful? So always remember, any F1 into recessive parent cross is called as test cross. Okay, it is called as test cross. So what how it will be helpful for us to know the genotype of F1 individual. We can get the knowledge about the F1 individuals. Okay? Yes. Next. Next. Yeah, next. Who proposed the chromosome theory of inheritance? So, who gave us chromosomal theory of inheritance? Their name was Walter Sutton and Theodore Boveri. Okay. Now, next question. Define true breeding. Mention its significance. So, what is true breeding and what is its significance? Okay. So, production of homozygous progeny through repeated self pollination. Okay, through repeated self pollination is called as true breeding. Okay, so when we are producing any homozygous progeny and it is by what, by self pollination, so that is called as true breeding. Okay, so what is its significance? How is it helpful? So we can transfer our desirable character. Our desirable character can be transferred from several generation. Okay, several generation without segregation, means without any changes. Okay, now next question. Explain the term phenotype and genotype. What do you mean by phenotype? Phenotype is nothing but physical appearance. Genotype is nothing but genetic makeup of an organism. Okay, next. What is point mutation? What do you mean by point mutation and give any example? Next, M- mutation due to change in single base pair. Okay, single base pair of DNA is called as point mutation. Example, sickle cell anemia. Okay, sickle cell anemia is caused by this mutation only. Next question. Our next sixth question is, yeah, our sixth question is, what will be the phenotypic ratio of the offspring obtained from the following crosses? Now, what will be the phenotypic ratio? Okay, appearance, phenotypic. If it is capital A and small a, the phenotypic will be 1:1. Means one will be tall, one will be dwarf. Next, if both are in homozygous conditions, one dominant and one recessive, so there will be no dwarf plant or recessive trait. Everyone will be a dominating trait. Okay, next, capital is small a, capital is small a. So 3:1. That means three of the offsprings will be tall and one will be short. Okay, next, capital A small a into capital A capital A. So there will be no dwarf plant and only tall plants, or we can say only the plants which will show dominate character. Okay, next here in garden P, the gene T for tall is dominate over allele for dwarf. Obviously, give the genotypes of the parents in the following crosses: A tall and dwarf, producing all the tall plants, and B tall into tall, producing three tall and one dwarf plants. Okay, so for first case, where I have to get all tall plants, but I need to cross what? One tall and one dwarf. A tall into draft, and this draft, so which type of draft you will take? Is it a heterozygous condition or a homozygous condition? So in which condition we get all the plants tall? So obviously if completely homozygous condition, if means one complete homozygous condition tall and one recessive trait which is a short draft, and if we cross them, all the progeny, that means F1 generation will completely will be tall. Okay, so in B, what we have, we want three tall and one dwarf. So we need to use heterozygous parents. We have to use heterozygous parents. Okay, then only we are able to get uh, three tall and one dwarf. Okay, so these are the short questions that can be asked in this chapter. Okay. Next, let us talk about molecular basis of inheritance. Okay, molecular basis of inheritance. Now, distinguish between heterochromatin and euchromatin. Which of the two is transcriptionally active? So, what do you mean by heterochromatin? It is a darkly stained and densely packed part of chromatin. Okay, so the dark part and densely packed is called on chromatin. It is called as heterochromatin. Euchromatin is nothing but lightly stained and loosely packed. Okay, so what which one of two is transcriptionally active? So to show transcription. What do you mean? Show transcription is nothing but protection of RNA, right? So euchromatin is transcriptionally active. To show the transcription, euchromatins are active compared to heterochromatin. Okay, next. Who proved that DNA is genetic material? What is the organism they worked on? So their name were Alfred Hershey. Okay, he and Martha Chase. Hershey and Chase experiment we call, right? So these both scientists, they explain and they told that DNA is a genetic material in the year 1952. Always remember, years are very important. You have to remember the years. Okay. Next, on bacteria, on bacteriophage. So which organism did they perform the experiment? On bacteriophages. Okay. Next, what are the components of a nucleotide? So what are the components which are present in nucleotide? Components are, there are three components. Nitrogen is base, where we get all our purines and pyrimidines. Next is pentose sugar and phosphate group. These three elements combine together to form a nucleotide. Okay. Next question. 5 AATGCAGATC. This is a sequence which is a given. So write the sequence of its complementary strands. You need to write the complementary strand, obviously, which is completely opposite to. If we have G, we need to write C. G means C. If A, we have to write T. Okay, and the mRNA. So what mRNA we are going to get? So for this, now it is very simple. Instead of just looking, just remember, just take this strand and you can write as it is here. Just you remember one thing, in place of T's, wherever we have T, you need to write U, because we know thymine is absent in RNA, and instead of thymine we have uracil in the RNA. That's it, nothing else. Okay, nothing else to remember for mRNA sequence. Okay, now name any three viruses which have RNA as genetic material. Very important. So, what are the viruses which have RNA as genetic material? You need to remember tobacco mosaic virus, QB bacteriophage, and HIV. Okay, these three viruses have RNA as the genetic material rather than DNA. Okay, next question. What are the components of transcription unit? So for transcription, what are the components do we require? First, a promoter site, second, the structural gene, and third, a terminator. Okay, a promoter, a structure gene, and a terminator. So I hope everyone remember the name. Here we will have promoter, here we have terminator, and this middle part is called as structural gene. Okay, next. What is the difference between exons and introns? Okay, so what are the difference between exons and introns? Exons are coding sequence that appear in mature or processed mRNA. Yes. So exons are coding sequence which are only appearing in matured or processed mRNA. If the RNA is not mature, I mean just now the mRNA came out of a DNA and it is not a mature RNA. So that RNA doesn't contain any exon. They will only contain introns. Okay. Next, introns are non-coding sequence and are not appear in mature or processed RNA. Obviously, if it is mature, we don't have introns. If it is mature, we only have exons. Okay. Next, what is meant by capping and tailing? This capping and tailing are generally seen where during a lacoperon. Okay, so what do you mean by capping? Addition of methyl guanosine triphosphate at the end of 5 prime end in HNRNA. This is called as capping. What are you telling? Addition of adenylate residues. Okay, at 3 prime end of HNRNA. This is called as tailing. Okay, tailing and capping. Very important. Which element we are adding and what end of the RNA is very important. You have to remember these questions. Okay, next. What's meant by point mutation? Just now we have learned, right, in like molecule also, the previous chapter. So change in a single base pair of DNA is called point mutation. So what do you mean by point mutation? Change in a single base pair of DNA is called as point mutation. Example, same sickle cell anemia. Okay, next. Next. What is meant by charging of tRNA? So there is a term called as charging of tRNA. What do you mean by that? Amino acids are activated in the presence of ATP and are linked to their related tRNA. This is known as charging of tRNA. Okay, so activation in the presence of ATP and they are linked to their related tRNA. This is called as charging of tRNA. Okay. Next, define stop codon. What do you mean by stop codon? Write the stop codon. Define stop codon and write the stop codon. The codon of mRNA which not code any amino acid helps to stop the translation is called as stop codon. See, once the what we say amino acid chain start forming, at some point we have to stop that formation, right? So to stop that formation, we have some stop codons, and these codons generally don't code for any amino acid. Okay, any amino acid. So that's why it is called as stop codons. What are the stop codons? UAA, UAG, and UGA. Okay, very important. Next, write any write any two chemical differences between DNA and RNA. What is the difference between DNA and a RNA? Chemically, we have to write. Okay, you can say, ma'am, DNA is double helix, RNA is the same, RNA has only single strand. But no, I'm asking you chemically, how is it different? So obviously, in DNA I have thymine, but in RNA I have uracil. Very obvious. Here we have deoxyribose sugar, whereas here we have ribose sugar. Okay, chemically this is different. Okay, next. So next chapter we can learn principle and process. Okay, principle and process for biotechnology. Okay, now what are molecular scissors? Where are they obtained from? So, what are molecular scissors? Restriction endonucleases are called as molecular scissors due to their ability of cleavage of DNA molecule. Cleavage means, see, if DNA is this double strand, no, this DNA polymerase, it can go and it can break, it can, make a gap between this DNA so that transcription, DNA replication can happen. Okay, replication we say, that process. So that's why they are called as molecular scissor also. Okay, why are they okay, so generally we can see them in bacteria. Okay, name any two artificially restructured plasmid. So we know what are plasmids. We know what are plasmid. Plasmids are nothing but extra chromosomal DNA, right? So, but artificially also we have made some plasmids. So what are they? PBR 322, PUC 19, PUC 101. Okay, these are the three artificial restructured plasmids. Next, what is EcoRI? How does it function? EcoRI is a restriction endonuclease enzyme obtained from E. coli. From E. coli bacteria, we get our EcoRI. Okay, it recognizes. So, how this EcoRI recognizes? So, it will recognizes this part. This is sequence. Okay, only this sequence it is going to recognize. Palindromic sequence and cut the DNA in between G and A and form sticky ends. So palindromic means even if you read from last to front and front to last, it should be same. Similar means it will be similar. Okay, palindromic sequence. Okay, next. What are cloning vectors? Give an example. So vectors are used for multiplying the foreign DNA sequence are called cloning vectors. So vectors which are used to multiply the DNA sequence, those are called as cloning vectors. Example, plasmid and cosmids. Plasmids and cosmids are the very good example for this. Okay, next. What is palindrome sequence? What do you mean by palindrome sequence? The sequence of base pair of DNA that reads same on the two strand, then orientation of reading is kept the same. Same palindromic. Palindromic sequence means two strands which reads the same. Okay. Now, what is the full form of PCR? How is it useful in biotechnology? PCR means polymerase chain reaction. This polymerase chain reaction recently also it got very populated for our what? COVID-19 test. Yes. COVID-19 test was also taken by using this PCR techniques only. Polymerase chain reaction. Okay, so in biotechnology, how is it useful? Amplification of gene of interest means if I want a particular strand, my desired strand, okay, and I see if I have only one strand, it is not useful, right? I need multiples. So to multiply this desired DNA or gene, what I can use? I can use PCR. That is nothing but polymerase chain reaction. Okay, next. What is downstream processing? What do you mean by downstream processing? Separation and purification of foreign gene product in biotechnology is called as downstream processing. Yes. So what do you mean by this? Is see, I have a bunch of DNA. So in this bunch, my desired DNA gene will be something else. So in this, whatever the separation I am doing, whatever the purification I am doing in this DNA, in complete DNA, and I am just extracting the my desired gene, that is called as, that is product gene product, that is called as downstream processing. Okay. Next, how does one visualize DNA on an agar gel? So how, see, agar gel is very much useful in what we say, electrophoresis. Okay, so in this electrophoresis, how this agar gel is, this DNA is visually, I mean, we can visualize in agar gel. So staining of DNA with ethidium bromide followed by exposure to UV radiation gives bright orange color to separate DNA bands. So, see, DNA strands will get stained, and these stains can be given by ethidium bromide, and we can and after treating after staining it with ethidium bromide, we can expose it to UV light. So when we are exposing it to UV lights, what happens? It will start showing us some colors. So with the help of that colors, we can see the DNA, or we can separate the DNA. Okay. Now, how can you differentiate between exonuclease and endonuclease? So what do you mean by exonuclease? These enzymes remove nucleotides from the ends of the DNA. Okay. Palindromic sequence are not required. Okay. Next, endonucleases, these enzymes cut the DNA as the specific position. Specific position here at the ends. Here, specific position it they want. Okay, so palindromic sequence is required for this endonucleases. That means I can say that what we say, eco-RI, it's a endonuclease, not exonuclease, because this eco-RI needs a what? Particular sequence to cut the DNA. Okay, next. Yeah, diagram of PVR 322 is very important. Okay, keep that in your mind when you are going through this chapter. Okay, biotechnology and its applications. Now, give different types of cryogene and pest which are controlled by protein encoded by this gene. So, cryogene, um, cryogene, where we are using this and how many types of cryogens we have. So the proteins of cryogene 1 AC and cryogene 2 AB control cotton bollworm. Okay, so in cotton, if we have some bollworms, these can be controlled using this cryogens. Okay, next, the protein of cryogene 1 AB control con. So different types of conjugate, how is it useful? Okay. Okay, next. Okay. Next question. What is GEAC and what are its objective? So what are its objective? Now, GEAC, Genetic Engineering Approved Committee, make a decision regarding the validity of genetic modified and the safety. So whatever the genetic modification is taking place, that will be taken, that will be analyzed, validated. We have to give some certifications, right? So that will be given by GEAC only. Okay. Next. Next question. Name the nematode that infects the root of tobacco plant. Name the strategy adopted to prevent this infection. So, which type of nematode will infect the root of the tobacco? It is Meliodigine incognita. Okay, and how it is? What is the strategy? It will interfere the RNA. Okay, it will interfere in the RNA. Okay, so these are very important questions which you have to go through. Okay, and I hope these questions will be useful for you as well. Okay, we have one Okay, two more questions. We will just finish it off. Okay. Give one example for each of the transgenic plant which are suitable for food processing and those who will improve the nutritional quantities. To improve the nutrition and how is it useful? Transgenic plant suitable for food processing is flower sour variety of tomato. Okay, in tomatoes. Transgenic plant with improved nutritional value is Typhi golden rice with vitamin A rich nature. So this golden rice, why is it very useful? Because it can be grown in any temperature with even adequate water also we can grow golden rice. So that's why it is very very good. What we said, transgenic plant. Okay, next. What is green revolution? Who is regarded as father of green revolution? So what do you mean by green revolution? The improvement of yield of crops through convention and non-conventional breeding methods to meet the requirements of human population across the world is called as green revolution. So in the agriculture method, in the agriculture field, to improve the yield of crops, okay, to improve the yield of crops, and as the population are increasing, obviously demand are increasing. So to increase the increase as the demand is increasing, we need to increase the yield also. So this yield production, like growth in this yield, this is called as green revolution, and this green revolution is given by N.E. Borlaug. Okay, N.E. Borlaug. Okay, so that's it, dear. So for today, these three chapters are enough. Question answer of these three chapters are well enough. Okay, next I'll meet you in the next class and we'll continue our next concept. Okay, so I'll see you